Properties of genetic material, DNA versus RNA. Now we are going to see why DNA is more capable of being a genetic material than RNA. After the experiment of Frederick Griffith, it was established that the genetic material, also referred to as transforming principle, is capable of transferring from one bacterium to another and alter the entire function of an organism. But for a long time it was believed that it is protein that acts as genetic material due to its extensive functional importance. It was only after Hershey and Chase experiment it was unequivocally proved that DNA is the genetic material and not protein. However, it subsequently became clear that in some viruses such as tobacco mosaic virus, bacteriophage, RNA is the genetic material. Automatically, a question would arise. Why DNA is the predominant genetic material, whereas RNA performs dynamic functions of being a messenger and adapter? The answer lies in the difference between the chemical structure of the two nucleic acid molecules. But before that, let us come to know about the properties that are required for a molecule to be a genetic material. So, a molecule that can act as a genetic material must fulfill the following criteria. First, it should be able to generate its replica. Only if it undergoes the process of replication, it would be able to pass on the characters from parents to progeny. Next, it should be chemically and structurally stable. It should provide scope for slow changes, mutation that are required for adaptation and evolution. Adaptation is a very important characteristic for a genetic material. Only then the organism will be able to survive. Next, it should be able to express itself in the form of Mendelian characters. Now let us see whether it is DNA or RNA which can fulfill these characteristics more satisfactorily. So coming to the first criteria, it should be able to generate its replica. This is important because only if a genetic material is able to replicate, it will be able to pass on the genetic information from parent to progeny. Because of the rule of base pairing and complementarity, both the nucleic acids, that is DNA and RNA, have the ability to direct their duplications. But then if you consider other biomolecules such as proteins, it fails to fulfill the first criteria itself. Therefore, only DNA and RNA have the capability to be the genetic material. Next criteria is that it should be chemically and structurally stable. This is again very important because only if a genetic material is stable, it will be able to pass on the genetic information from one generation to next generation with efficiency without causing much alteration in the genetic setup. So DNA that has two strands being complementary, if it's separated by heating or any other cause, it can come together when appropriate conditions are provided. Therefore, DNA has evolved a mechanism known as repair, by means of which it can become functional again even if it is destroyed. And then the plane of one base pair stacks over the other in double helix. This confers additional stability to the helical structure of DNA. So the presence of 2-OH group at every nucleotide in RNA is a reactive group. Therefore, it makes RNA liable and easily degradable. The presence of thymine at the place of uracil also confers additional stability to DNA. And then RNA is also known to be catalytic. Therefore, it is highly reactive. You can see that DNA has evolved a process known as repair, and then it has additional stability since the plane of one base pair stacks over the other. And then it has thymine at the place of uracil, which also confers additional stability to DNA. Therefore, you can say that DNA is chemically less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, DNA is a better genetic material. So next is that it should provide scope for slow changes, that is mutation, that is required for evolution. So both DNA and RNA are able to mutate. Okay. In fact, RNA being unstable mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve fast. Mutation should not be extremely predominant in a genetic material because higher mutation can alter the genetic information it carries, and hence the genetic setup cannot be efficiently passed on to the progeny. Mutation only for the sake of adaptation proves to be advantageous. So DNA seem to provide the sufficient mutation that is required for adaptation. So next characters is that it should be able to express itself in the form of Mendelian characters. RNA can directly code for the synthesis of proteins and hence can easily express the characters. But DNA, however, is dependent on RNA for synthesis of proteins. So you can see that the protein synthesizing machinery has evolved around RNA. Suppose if RNA is a genetic material, there is no need for DNA, and the storage becomes problematic because of its high reactivity and tendency to undergo mutation. But if DNA is a genetic material, since it's dependent on RNA for protein synthesis, it is kind of ensured that all the three biomolecules plays its important role. So you can see that DNA being more stable is preferred for storage of genetic information. But then for the transmission of genetic information, RNA is better. So since the lifetime of organism has increased during the course of evolution, the genetic material needs to be accurately stored than being transmitted. Therefore, DNA with better storage capacity proves to be a good genetic material. Hence, DNA is the genetic material in majority of the organism. So now a question would automatically arise. Which is the first genetic material, whether it's DNA or RNA? We know that DNA is the genetic material in predominantly most of the organism, but RNA do act as genetic material in some viruses such as QB bacteriophage, tobacco mosaic virus and others. But which among these two was the first genetic material to have evolved? Now there is enough evidence to suggest that RNA was the first genetic material. So RNA used to act as genetic material as well as a catalyst. So these two are most important biomolecules in a living system. So there are important biochemical reactions in the living system that are catalyzed by RNA catalyst and not by protein enzymes. So essential life processes such as metabolism, translation and splicing also evolved around RNA. So translation, if you consider, the production of proteins takes place from RNA, right? And splicing also takes place in RNA, as we are going to see in the next few segments. But then RNA being a catalyst was reactive and hence unstable. There are many other factors which decrease the stability of RNA and made it reactive. But the genetic material should be stable for it to be able to transmit the genetic information from parents to offspring. So therefore DNA has evolved from RNA with chemical modifications that makes it more stable, like the presence of thymine at the place of uracil and in the double helical structure, the plane of one base pair taking over the other, and many other modifications. So DNA also being double stranded and having complementary strand further resisted changes by evolving a process of repair. Hence, DNA was able to be a better genetic material than RNA. Therefore, even though RNA was the first genetic material, DNA being more stable became the genetic material in majority of the organism, mainly because of its ability to store the genetic material accurately without much changes. And it also allows mutation only at a very slow rate, ensuring that there is efficient transformation of information from parent to progeny to be able to carry over the whole genetic setup through generations.